Biodegradation of xenobiotic pollutants. A xenobiotic is a chemical substance found within an organism that is not naturally produced or expected to be present within the organism. This is something foreign substance which a person or individual consumes and that is not present naturally, so the system is not having any metabolic pathway to utilize that. These xenobiotic compounds include pharmaceutical drugs like aspirin, which is used as a pain reliever and anti-inflammatory drug, antibiotics like penicillin, ciprofloxacin, or chemotherapeutic agents which are used in cancer treatment. Also, pesticides and herbicides are considered as xenobiotic compounds. They include DDT, which is a banned insecticide as it gets accumulated, glyphosate, which is a common herbicide, malathion, which is organophosphate insecticide. Also, some industrial chemicals and pollutants are considered as xenobiotics. They include PCBs, that is polychlorinated biphenyls, which are used in electrical equipment and they are now banned. Also, dioxins, which are byproducts of industrial processes, and benzene, which is used in the chemical manufacturing and which is carcinogenic. All these are xenobiotic compounds. Some food additives and preservatives are also xenobiotic, like sodium benzoate, which is most commonly used as a preservative in soft drinks, artificial sweeteners like aspartame, which are used by diabetic patients, monosodium glutamate, which is a flavor enhancer. Then there are some chemicals which are used in cosmetics and which are also personal care ingredients that include parabens, phthalates, and triclosan. All these are also xenobiotic, that means they are not present naturally in the system. Parabens are mainly used as preservatives in shampoos and lotions. Then phthalates are used to soften plastics and also they are used for fragrances. Triclosan is antibacterial agent which was being used in soaps. There are some environmental contaminants like microplastics, which are tiny plastic particles present in water and food. They are also present in chewing gums. Bisphenol A, BPA, which is found in plastics and linings of cans, and heavy metals like lead, mercury, and cadmium. All these are examples of xenobiotic compounds. These xenobiotic pollutants are mainly the result of industrial revolution and rapid urbanization. The global environment has been increasingly polluted by a complex mixture of these xenobiotic compounds. The examples of these xenobiotic compounds include azodyes, phenolics, polycyclic aromatic hydrocarbons, halogenated compounds, personal care products, pharmaceutically active compounds, pesticides, nitroaromatic compounds, triazides, and chlorinated compounds. The environmental hazards of these xenobiotic compounds mainly include their persistent nature. That means they take longer time or more efforts to get degraded. As a result, they get bioaccumulated. That means they will get accumulated or saturated in organisms and they get magnified in the food chain, which results in biomagnification. Also, some of these xenobiotic compounds are toxic. They may be teratogenic or carcinogenic or mutagenic. Historical concern rose with discoveries in 1960s of DDT and methyl mercury in wildlife and fish. The compounds which were used as insecticides or herbicides, they get accumulated in these organisms and they disturb the ecosystem. There are certain limitations of traditional remediation methods like coagulation, filtration, and electrolysis, as these methods are expensive, they are technically complex, they need some machinery or expertise, they demand space, and potentially these processes may be harmful as they may produce some toxic byproducts. Bioremediation is a sustainable alternative approach which can be used for degradation of these xenobiotic compounds. As biodegradation is eco-friendly, cost-effective, and sustainable approach. Role of microorganisms in xenobiotic degradation. A wide range of bacteria, fungi, and plants participate in breaking down these xenobiotic compounds. Notable microbial genera which are involved in these bioremediation processes include bacteria like alkaligens, Microbacterium, Micrococcus, Methanospirillum, Aeromonas, Sphingobiotum, Flavobacterium, and Rhodococcus. Also, fungi can be used for bioremediation of these xenobiotic compounds. The fungi include Aspergillus, Penicillium, Trichoderma, or Streptomyces. Some yeasts can also be used for this process. They include Rhodotorula, Candida, and Orobasidium. This diagram shows the microbial enzymes which are used for xenobiotic detoxification. The arrow indicates enzyme, for example, amylase and keratinases. These enzymes, which can be used for degradation of xenobiotic compounds, they are produced by certain fungi like phanerochetes, certain bacterial sources like Bacillus megaterium. Pectinases are the enzymes which are produced by Aspergillus niger, Pichia fermentis. Bacterial sources include Bacillus. Then for lacase, fungal sources are there. Also bacterial sources are there. Cellulases, cytochrome P450, lipases, proteases, phytase. All these are different enzymes which are produced by different fungi and bacteria. Xenobiotic degrading enzymes associated with bacteria. Bacteria exhibit remarkable capabilities for rapid proliferation and resilience. That means they get adapted to the adverse condition so that they survive and they can degrade these pollutants. Recent genomic analysis indicates that xenobiotic degrading bacterial strains have evolved through the acquisition of specialized catabolic genes. Various bacterial species, including Pseudomonas, Escherichia, Sphingobium, Rhodococcus, Bacillus, Gordonia, and others, have been isolated from soil environments and they have been characterized for their ability to degrade these xenobiotic compounds. They can degrade DDT, that is dichloro-diphenyl dichloroethane, lindane, polychlorinated biphenyls, trinitrotoluene, and crystal violet. Additionally, the human intestinal microbiota possesses intrinsic, that means inbuilt, xenobiotic metabolizing potential and modulates host gene expression and enzyme activity related to xenobiotic metabolism. For instance, a thermotolerant strain identified as Gordonia species was isolated from oil contaminated soil, demonstrating significant biodegradation capacity. Under anaerobic conditions, aromatic xenobiotics functions as electron donors, with microbial communities oxidizing these compounds in presence of appropriate electron acceptors. So due to the redox potential of these organisms or by certain enzymes produced by these organisms, the xenobiotic compounds can be converted to simpler molecules. The selection of suitable enzymes is critical for bioremediation, as these enzymes catalyze the transformation of hazardous xenobiotics into less toxic metabolites. The structural diversity of hydrocarbons necessitates distinct enzymatic pathways for their degradation. Same enzyme or same metabolic pathway cannot degrade all different molecules. So there is a need of different enzymatic pathways or different enzymes having different metabolic pathways. Aliphatic hydrocarbons undergo monooxygenase or dioxygenase mediated oxidation. These enzymes add one or two oxygen, leading to fatty acid formation. These fatty acid molecules are ultimately the part of TCA cycle. They enter TCA cycle, they produce carbon dioxide and water. And as a part of TCA cycle, they become energy yielding molecules for the organism. In contrast, Aromatic hydrocarbons are more recalcitrant due to their low solubility and toxic intermediate produced during their degradation. So aromatic hydrocarbons are difficult to be degraded. The initial step of these degradation pathways involve conversion to cis dihydrodiols, where two OH groups are added to adjacent carbon atoms, followed by ring cleavage, that is breakdown of the aromatic ring, or dioxygenase enzymes through either ortho or meta cleavage pathways, where the aromatic rings are getting cleaved to produce simple molecules, and ultimately these will yield intermediates that feed into central metabolic pathways, ultimately producing the energy molecules. This table gives the examples of certain xenobiotics, bacteria which are involved in degradation of these xenobiotics, enzymes produced by those bacteria, and mechanism of degradation. So aliphatic hydrocarbons like haloalkanes, including 1,2-dichloroethane, these are degraded by bacteria like Xanthobacter, then Rhodococcus. These bacteria produce enzymes like haloalkane dehydrogenase, and they give the mechanism of degradation by nucleophilic substitution reaction to catalyze displacement of chlorine from these molecules. Medium and long chain alkanes are degraded by Pseudomonas. The enzyme which is involved is alkane hydroxylase. The pathway involved is oxidation of the terminal, that is end carbon atom, yielding an alcohol. Sterols are degraded by Rhodococcus. Enzyme involved for this pathway is oxygenase. This oxygenase enzyme catalyzes hydroxylation, and possibly it further carries out the oxidation of C26 atoms of sterol. Aromatic hydrocarbons including azo-dyes are degraded by Ganoderma species by the production of enzyme lacase. This enzyme oxidizes phenolic and methoxyphenolic acids and decarboxylate them and attack their methoxy group. Estrogens are degraded by Pseudomonas putida strains by removal of organic substrate electrons and ultimately reducing dioxygen molecules. Xenobiotics like nitroaromatic compounds, including 2-nitrophenol, 4-nitrobenzoic acid, 2-nitrobenzaldehyde, and 3-nitrophenol, are degraded by bacteria Xenophilus, Enterococcus, and Geobacillus. Also, Pseudomonas can degrade these compounds by production of enzyme azoreductase. These enzymes, adoreductase, they carry out reduction of azo bonds. Catechol and chlorocatechol, they are degraded by Pseudomonas. The enzyme which is involved is chlorocatechol 2,3-dioxygenase. Catechol gets transformed into a ring cleaved product, that is 2-hydroxymuconic semialdehyde, and this will then again get cleaved between to form two different molecules which are hydroxy substituents of protocatechoic acid with the incorporation of molecular oxygen to form beta carboxymuconate. Same pathway is involved for protocatechoate. Acinetobacter is involved in degradation of that molecule by production of enzyme protocatechoate 3,4-dioxygenase. Polyaromatic hydrocarbons like naphthalene, they are degraded by Pseudomonas, different species of Pseudomonas, by production of enzyme naphthalene dioxygenase and ring hydroxylating dioxygenase. The pathway which is involved is oxidation of one of the aromatic rings of naphthalene using molecular oxygen. Xenobiotic compounds can also be degraded by the enzymes associated with fungi. Fungi can reduce pollutant concentrations not only through metabolic degradation but also by physically absorbing contaminants onto their thick cell walls, that is adsorption. which are composed of structural polymers such as chitin and cellulose. The decomposition of xenobiotic compounds by fungi underscore the critical role of their intracellular enzymatic systems in the transformation of these pollutants. That means fungi can give both the mechanisms. First, these compounds can get associated with the cell wall, and if they are absorbed inside the cell, then intracellular enzymes can degrade or transform these xenobiotic molecules. Fungal species contribute to a variety of sectors through their biodegradative capabilities, including biofuel production, environmental management, and industries such as food processing, paper making, beverage manufacturing, and textiles. This table gives the examples of xenobiotics which are degraded by fungi. Aromatic hydrocarbons like beta-lactam are degraded by Fusarium species by enzyme lactamases. It hydrolyzes an aromatic polyketide into endochrosin 9 anthrone. Atrazine can be degraded by the fungi by the production of enzyme lactamase or tyrosinase, manganese peroxidase, manganese independent peroxidase, and lignin peroxidase by dealkylation of atrazine, which results in fragments of aldehyde or ketone. Atrazine monocrotophos DDT. These xenobiotics can be degraded by Fusarium species by production of enzyme N-acetyltransferase and N-malonyl transferase. It helps in the detoxification and degradation of aromatic amines. Aromatic compounds, aliphatic hydrocarbons and PAH, they can be degraded by different fungi including Trichoderma, Penicillium, Aspergillus, different species of all these fungi. They produce enzyme lactase, manganese peroxidase, epoxide hydroxylase, cytochrome P450, monooxygenase, dioxygenase, protease, and lipase. And by peripheral degradation pathways of organic pollutants, they are gradually transformed and many intermediate products are formed which can ultimately enter the central metabolic pathways. Chlorpyrifos can be degraded by cladosporium, by chlorpyrifos hydrolase enzyme, pectin methyl transferase enzyme, and polygalacturonase enzyme. These enzymes are responsible for pectin degradation by catalyzing the demethoxylation of homogalacturon chain and pectin to release methanol and acidic pectin. Then lignin, PCBs, petroleum hydrocarbons, PAH, trinitrotoluenes, and other industrial di-effluents, herbicides, and pesticides can be degraded by different fungi by production of enzyme lignin peroxidase, versatile peroxidase, lactase, and manganese peroxidase. These enzymes help in formation of semiquinone intermediates during oxidation of lignin-derived hydroquinone by lactase. It cleaves carbon-carbon bond and oxidizes benzyl alcohol to aldehydes or ketones. Xenobiotics like nitroaromatic compounds can be degraded by phanerocheate by peroxidases that degrade various nitroaromatic compounds by initial reduction of nitro group to hydroxylamines. Navy blue HAR, indigo, and other azodyes can be degraded by fungi trichosporon by the production of enzyme lactase. It attacks phenolic subunits and degrade dyes leading to C-alpha oxidation, C-alpha beta cleavage, and aryl alkyl cleavage. PAH and PHC can be degraded by aspergillus by production of enzyme lactase and peroxidase by degradation of benzoalpha pyrene phenanthrin. So by all these different pathways, xenobiotic compounds or xenobiotic pollutants can be degraded by activities of different bacteria and fungi.